Okay. So good afternoon. Today morning we have seen in the first session about food safety concerns related to poultry meat and their products. So wherein we have discussed about the chemical hazards and microbial hazards that may occur in poultry meat in general. However, we have to have an insight into the microbial hazards, especially bacterial pathogens like Salmonella and Campylobacter. As a faculty of livestock products technology, I feel that if we provide a special insight into the bacterial pathogens like Salmonella and Campylobacter, that would be useful for the fellow faculty. So that I have incorporated this topic on microbial hazards in poultry meat, especially on Salmonella and Campylobacter. In this regard, I call upon Dr. K. Porteen to deliver his lecture. Dr. Porteen has served as a faculty, served as a postdoctoral fellow in Food Microbiology Laboratory, USDA, Maryland, for 4 years from 2007 to 2011. Thereafter, he has joined as assistant professor in Tamil Nadu Veterinary Animal Sciences University in Department of Veterinary Public Health and Epidemiology. Dr. Porteen, please take your time.
Good afternoon, one and all who are assembled for this online session. At the outset, I would like to thank the organizers for selecting me and giving me an opportunity to be a part of this wonderful training program. Thank you, sir. So my topic today is going to be on microbial hazards in poultry meat. It is going to be a general introduction as conveyed to me by the organizers. It is going to be a general talk. And here we are going to give special emphasis on two important pathogens. One is Salmonella, another one is Campylobacter. Myself, K. Porteen, Assistant Professor, Department of Veterinary Public Health and Epidemiology, Madras Veterinary College. Sir, am I audible, sir?
Audible, Porteen.
Okay, sir. Okay, sir. So the first slide is going to be on what is known as poultry meat. These are some known facts. I hope all the audience who have gathered over here will be knowing all these facts. This is a general information. This is just to refresh your memory. Going into our subject proper, this is just to refresh your memory. We'll talk about poultry meat. As you all know, poultry meat is a major source of protein for majority of the people who are living in low-income regions, such as Sub-Saharan Africa and South Asia. It is loaded with what is known as proteins, B-complex vitamin, and it is a good source of what is known as phosphorus and other minerals. As such, poultry meat is low or minimal in harmful trans fats, but it is high in what is known as beneficial monosaturated fats. Special category of people, namely pregnant women, children, and elderly people, derive maximum benefit by consuming what is known as poultry meat. Many studies in recent times have pointed out this poultry meat can make a significant difference and also contribute to the fight against what is known as child malnutrition. In other words, poultry meat is going to be a substitute for what is known as the war against child malnutrition. So these are some basic facts which you people will be knowing. This is just to refresh your memory. So in continuation to this one, we'll see about the poultry meat facts. Many studies has been proved that the per capita consumption of meat was highest in Northern American countries, which is approximately somewhere around 97.5 kilos per person in a period for about 2 years. Somewhere around 2018 to 2020, it has been identified that the maximum consumption was there in Northern America, which was somewhere around 97.5 kilos. In Africa, it has been identified an average person consumes somewhere around 13 kilos per year. So this is some nutritional fact which you are going to talk about. The meat of chicken contains somewhere around 3 grams of fat per 100 grams of meat, whereas red meat contains double the amount. It is somewhere around 5 to 7 grams of fat per 100 grams of meat. Among these red meat, mainly beef and mutton, they contain what is known as higher amounts of trans fat, but that is not the case with regard to what is known as chicken meat. Chicken meat contains more of what is known as valuable monosaturated fats, and it contains only less amount of this or less or minimal amount of this saturated fats. So as a result, this red meat predisposes human beings to various ailments like coronary heart disease, and it is very common in case of what is known as human beings. Like World Cancer Research Fund has postulated, it is not only World Research Fund, like World Cancer Research Fund, many other organizations have clearly pointed out that the consumption of red meat, somewhere around 500 grams per week, that particularly processed meat, may be unhealthy, but that is not the case with regard to what is known as poultry meat. Like as of now, India ranks 6th in the broiler meat production, but the consumption of this meat in countries like India is very poor. So owing to its cheap availability and loaded with nutrients, and this can be promoted, and this is happening in most of the developing nations. Now people are more fascinated towards consumption of what is known as all these poultry meats. So this is just to give an overall view with regard to the production of meat worldwide from 2016 to 2021. You can see that there is a steep increase in what is known as production of all these meat. Like you can see, like it was only 115 million metric tons in the year 2016. Now it has come to somewhere around 135 metric tons in the year 2021. So this is just to give an emphasis that the production of this poultry meat is on an increasing trend throughout the world. This is again a statistics which is being recently published. Like people have done some analysis and they have found that chicken meat production throughout the world it is on increasing trend, and it is not only the production, the consumption is also increased. It is on an increasing trend. Like you can very well visualize. Like you can see in 2021, the consumption is somewhere around 133 metric tons, whereas if you see by 2030, the consumption is going to increase by another 20 metric tons. It will be somewhere around 151 metric tons. So this gives an emphasis, yes, that the meat production has increased. In a similar fashion, the consumption rate is also increasing. So this is just to say, like India ranks 6th in broiler meat production, and this is the latest statistics which has been seen in the year 2021. India ranks the 6th position in what is known as your poultry meat production. So we have seen what are the facts about all these poultry meat, like what is the importance of this poultry meat. We have seen all these things. Whenever there is an advantage, there is going to be what is known as some kind of disadvantage is there. As you all know, this poultry production, it is one of the fastest growing livestock subsector. There is an increase in what is known as production, but there is also increase in what is known as number of health issues. It is not only to the people who are working with what is known as poultry industry, but also to the people who are going to consume these poultry and poultry products. So mainly these infections are going to occur when you're going to consume and you're going to handle what is known as contaminated all these poultry meat. It leads to food poisoning throughout the world. With regard to most of the foodborne diseases, people have postulated that 70% of the foodborne diseases are of bacterial origin. That too, with regard to foodborne diseases, the two main pathogens which causes human health problems is Salmonella and Campylobacter. In recent times, there is a development of what is known as antimicrobial resistance with regard to all these microorganisms. It is a threat. It is a major threat to what is known as human health, and it has been attributed inappropriate use of antimicrobials in poultry production chain, like maybe a reason for development of all these antimicrobial resistant microorganisms. And moreover, this threat, it has been estimated by 2050 there is going to be 10 million deaths every year annually only due to this antimicrobial resistance. That is the importance of what is known as this emergence of all these antimicrobial resistant microorganisms. So as you all know, like presence of all these microbial hazards, like coupled with the antimicrobial resistance, it makes the product as well as food safety a big question mark. And these microbial hazards act as an important risk factor, and it tends to cause what is known as various ailments in case of what is known as human beings. With regard to hazards in poultry meat, as like all these audience will be knowing, like an hazard can be a physical agent, or it can be a chemical agent, or it can be a biological agent, like which has the potential to cause what is known as illness or injury. That can, like, unless until a control cannot be applied, that is known as what is known as an hazard. In general, there are three types of hazards are there: physical hazards, chemical hazards, and biological hazards. You'll be knowing, like physical hazard includes all these glass particle, packaging material, all these things comes under physical hazards. With regard to chemical hazards, pesticides, biocides, food additives are examples of what is known as chemical hazards. With regard to biological hazards, most of the microorganisms falls under biological hazard. That is what we are going to discuss for our today's lecture. With regard to microorganisms in poultry meat, we broadly classify all these microorganisms that is present in our poultry meat into two broader categories. The first one is going to be spoilage microorganisms. Coming to the spoilage organisms, the first and foremost thing should be kept in mind is these spoilage organisms are non-pathogenic in nature. That means they are not going to cause any kind of human disease, or they are not harmful to what is known as human beings. But these organisms has an inherent ability to multiply under what is known as lower storage temperatures, where they tend to produce what is known as off odors and off flavors, like which needs what is known as monitoring at various stage of processing, even at the time of what is known as storage as well as handling. And classical example of spoilage organism is going to be Pseudomonas. Like Pseudomonas is an example of spoilage organisms. Most of these spoilage organisms are psychotropic in nature. Like they are non-pathogenic, but they tend to cause what is known as some kind of off odors and off flavors, which leads to what is known as rejection of the project. That is the importance of what is known as the spoilage organisms. The second important category we are going to talk about is known as these foodborne pathogens. Like as the name indicates, these organisms can be acquired through what is known as food, and they are very commonly seen in what is known as poultry and poultry products. So whenever people talk about foodborne pathogens, people always restrict themselves to what is known as gastrointestinal disorders, such as diarrhea, vomition, all these things. Like apart from all these gastrointestinal disorders, these organisms has the ability to cause what is known as several systemic and chronic diseases such as abortion, stillbirth. Take for example listeria. Listeria has an ability to cause what is known as abortion. And listeria as what is known as the central nervous system involvement disorders, meningitis, encephalitis. Take for example, your Campylobacter has the ability to cause what is known as some kind of neuronal dysfunction, arthritis, like allergies. All these things will be caused by what is known as these foodborne pathogens. But whenever we talk about foodborne pathogens, we always restrict them to what is known as gastrointestinal disorders. Like apart from gastrointestinal disorders, these organisms has the inherent ability to cause both systemic as well as chronic diseases. So this is the broader classification of what is known as microorganisms that is present in meat. We classify them into two broader categories, namely foodborne pathogens as well as spoilage organisms. Spoilage organisms are non-pathogenic, and they will not cause any kind of disease manifestation in case of human beings, but they tend to cause what is known as the aesthetic quality of the meat will be affected. Whereas with regard to foodborne pathogens has the ability to induce what is known as disease in case of animals as well as in humans. The classical examples are Salmonella and Campylobacter. So why, like among the various foodborne pathogens, why importance is being given only to Salmonella and Campylobacter? Because this poultry acts as an important asymptomatic carrier as well as a reservoir of what is known as this Campylobacter as well as Salmonella organisms. That means the bird will be harboring the pathogen, but it is not going to exhibit any kind of disease manifestation, or it will not show any kind of symptoms, but continuously the pathogen will be excreted via feces. That is why this Salmonella and Campylobacter are very important when it comes to poultry meat, because poultry acts as both as carrier as well as the reservoir. And at the time of slaughtering, you'll be knowing, like whenever these gastrointestinal contents, when they come into contact with your meat, there is going to be what is known as contamination. That is one thing. And it has been noticed that these organisms persist in the nature. That means they are present in the environment. As a result, there is every chance of what is known as cross contamination that is there. And nowadays we always go for what is known as intensive system of rearing. There is more demand, so people go for what is known as intensive system of rearing. So in this intensive system of rearing, there is always close contact with the birds. So there is every chance of what is known as contamination. That is why Salmonella and Campylobacter are a major concern when it comes to what is known as poultry meat. So with this, we'll go into individual species. We'll talk about Salmonella, we'll talk about Campylobacter, then we'll see the control strategies, then we'll conclude. So this is how I'm going to proceed for this class. So coming to Salmonella species, like I don't know whether I don't know the background of the audience, since many may not be exposed to all these pathogens. So we'll discuss about the preliminary things about this pathogen. So this pathogen was identified way back in 1855 by Theobald Smith. At the time, people were thinking that this organism was responsible for causing what is known as hot cholera or classical swine fever. So many of the audience will be knowing that hot cholera or classical swine fever is caused by a virus disease. At the time, people were thinking But because this organism has been isolated from animals which were suffering from what is known as R. cholera or classical swine fever, a secondary bacterial infection, and people have attributed this may be the etiological cause for what is known as this R. cholera. Later, with the advance of what is known as science, people have identified like it was only a secondary bacterial infection that was happened due to what is known as Salmonella. So at the time when this Theo Paul Smith identified these organisms from pigs which were suffering from R. cholera or classical swine fever, there was one gentleman by name Dr. Salmon. He is an American veterinary pathologist. Like he identified this bacterial strain. So to honor Daniel Salmon, this pathogen was named as what is known as Salmonella. So this is the brief history about what is known as Salmonella. So coming to the classification of the Salmonella, like Salmonella comes under the family Enterobacteriaceae. Like in the genus Salmonella, there are only two species are there. One is Salmonella enterica, another one is Salmonella bungori. Like this Salmonella enterica is further subdivided into 6 subspecies, namely Salmonella enterica, subspecies enterica, Salmonella enterica, subspecies salame, Arizona, Di-Arizona, Hauten, and Indica. So this Salmonella enterica, this is the one which is going to harbor most of the pathogen which has the potential to cause both human as well as animal diseases. Whereas Salmonella bungori, like most of the species, like most of the serovars which is going to fall under Salmonella bungori, they are non-pathogenic in nature. Till now, WHO has identified somewhere around 2,500 serotypes. To number it specifically, like 2,659 serotypes have been recognized by WHO Collaborating Center for Salmonella Serotyping. Like an important thing has to be mentioned with regard to the Salmonella enterica subspecies enterica is like most serovars of both human as well as animal importance. That means the serovars which has the potential to cause infection in both animals as well as animals in case of humans falls under Salmonella enterica subspecies enterica. Like it has been identified till now, like 99% of the infection which is occurring due to Salmonella in both animals as well as in humans fall under Salmonella enterica subspecies enterica category. Like this is the recent classification. Like people have done like you will be naming Typhimurium, Typhi, Paratyphi, Newport, Dublin. All these things are not species; they are mere serovars. They fall under Salmonella enterica subspecies enterica. Under this one, they are all being placed, and they are not species; they are mere serotypes. So, like with regard to this serotyping, like classification up to subspecies level, it is mainly done by phylogeny, that is by DNA hybridization studies. Like people have gone for what is known as this subspecies classification. Whereas when it comes to serology is mainly based on what is known as 3 antigenic determinants, namely your somatic antigen, capsular antigen, and flagellar antigen. Like many will be knowing what is somatic antigen, capsular antigen, and flagellar antigen. These are various antigenic determinants that is present in the bacterium. Based on that one, like we have classified them into what is known as various serotypes. Based on this classification only, we are telling like till now we have somewhere around more than 2,500 serovars are present within this Salmonella group. So coming to the prevalence of this organism in poultry meat, a study has been conducted by CDC, and CDC has clearly pointed out of all the various foodstuffs, like you can identify 29% of the poultry meat is going to harbor what is known as this Salmonella organisms, and 18% of the egg is going to harbor what is known as this Salmonella organism. Remaining is being shared by what is known as other food sources, namely pork, beef, vegetables, fruits, nuts, and other food products. You can see, like with regard to poultry and poultry meat, the majority of the infection normally occurs through what is known as this Salmonella organisms. Like recent USDA report is like as pointed out, 3 important serovars are very important with regard to Salmonella, and they have the potential to cause what is known as the salmonellosis, that is Salmonella typhimurium, Salmonella enteritidis, and Salmonella heidelberg. These were the top 10 serovars which are commonly identified in poultry as per USDA reports. Coming to India, like in India again, the scenario is the same. Like non-typhoidal Salmonella, they are going to predominate. It has been found that chicken meat samples can contain as high as 40% of Salmonella, followed by poultry feces, which contains somewhere around 20% prevalence is being recorded. Coming to Indian context, in India, like 3 more common serotypes that has been identified with like from poultry meat are Salmonella Kentucky, Salmonella virchow, and Salmonella typhimurium. Recent studies have pointed out there are only 3 important serovars that is being circulated with regard to Salmonella that has been isolated from what is known as poultry meat. That one is Salmonella Kentucky, another one is Salmonella virchow, and the third one is Salmonella typhimurium. Coming to the Salmonella transmission from poultry, like as many will be knowing, the transmission can occur both through horizontal route as well as through vertical route. Like as you all know, like vertical transmission means from the parent to offspring is known as vertical transmission. Like with regard to poultry birds, one serovar is very specific with regard to what is known as a vertical transmission, that is serovar enteritidis, because this serovar enteritidis has a preference for what is known as chicken reproductive system. As a result, this vertical transmission is a great concern. Like what is happening is this bacteria, they tend to migrate from the cloaca to what is known as reproductive organs, and thereby this organism gain entry into what is known as intact eggshells. That is, since the organism is going to migrate itself into reproductive organs, like there will be, this organism is normally found in what is known as intact egg. That is the importance of what is known as this vertical transmission. But apart from this vertical transmission, the main mode of transmission that is happening in poultry in India or as well as in developed nation is horizontal transmission. That takes place through both aerogenous as well as fecal-oral transmission is there. Like we can very well say, like Salmonella is clearly transmitted horizontally by fomites, polluted drinking water, polluted feed, dirty cages, carrier birds, and feces of what is known as infected birds. And this is the primary route by which the organism is going to gain entry, and it is going to cause what is known as disease. Like this picture clearly depicts, you can see that when an animal is going to defecate, it comes to the plants, it comes to the water source. From there, human beings can pick the infection, or human beings can pick the infection directly from the birds. Like like the cycle is being recycled. That means it goes from humans to animal, it goes to environment. Again, from the environment, it is picked from the animal, from the bird. Again, from the bird, the cycle is keep on repeating. That means there is an intimate like relationship between the host, agent, and the environment. Thereby, the organism is able to survive till now. So coming to Salmonella pathogenesis, this is the general pathogenesis of Salmonella with regard to human beings. You can see like what happens is along with the food, you are going to take the organism, or the person is going to consume the organism. Like the organism has to reach the intestinal tract to elucidate what is known as the disease manifestation. Whenever the organism goes inside our intestinal tract, 3 things has to happen. One is addition and invasion of what is known as mucosal cells. That means the organism tends to adhere itself to what is known as this microbial. After addition, there is a phenomena, something that is known as a membrane ruffling. Like membrane ruffling is a phenomena that facilitates what is known as internalization of this pathogen. So once the pathogen is being internalized, like what happens is this pathogen tends to grow inside the vacuole. Imagine it is like a balloon. It is like a balloon. Like if you keep on pumping air into a balloon, at one point of time the balloon will burst. In a similar fashion, like here what happens is after internalization, the pathogen will be continuously multiplying inside the vacuole. At one point of time, there will be something that is known as lysis. So whenever there is going to be what is known as lysis, there will be tissue damage. To counteract this tissue damage, like all your inflammatory cells will come to that to the area of what is known as tissue damage. Once these inflammatory cells are going to come, they tend to release what is known as prostaglandins. So these prostaglandins, what it does is they try to elevate the level of what is known as the cyclic AMP. This when cyclic AMP is going to be elevated, there will be what is known as the electrolyte imbalance. That electrolyte imbalance, there will be increased chlorine secretion, and there will be reduced sodium uptake, which results in what is known as electrolyte imbalance that is manifested in the form of what is known as diarrhea. So this is the general pathogenesis of what is known as salmonellosis, which is going to happen in case of what is known as human beings. Like along with the food, we are going to take the organism is going to invade, it's going to multiply, and over the period of time there will be rupture. So whenever there is going to be tissue damage, there will be inflammation. Once inflammation will be there, prostaglandins will come in. These prostaglandins, they will elevate the levels of cyclic AMP. So electrolyte imbalance will be there. That electrolyte imbalance will be exhibited in the form of what is known as diarrhea. So this is with regard to what is known as general pathogenesis of what is known as Salmonella. So what are the various factors that is going to aid with regard to pathogenicity and virulence factors is, like in Salmonella, there is something that is known as the Salmonella pathogenicity island. The Salmonella pathogenicity islands are gene clusters. They are located in what is known as certain regions in the chromosome of the bacterial cells, and they are responsible for encoding various virulence factors like addition, invasion, and production of what is known as toxin genes. As I was discussing, To elucidate or to initiate the pathogenesis, the organism should invade, the organism should multiply, then it should have some toxic gene, then it is going to elucidate this disease manifestation. So all these pathogenicity and virulence factors, they are located with regard to what is known as Salmonella pathogenicity island. They are gene clusters. They are normally seen in what is known as chromosomes. Like various virulence factors which aid in the pathogenesis of Salmonella or Flagella, capsule, plasmids, adhesion system, and type 3 secretion system. And various virulence factors, they are commonly seen in what is known as Salmonella pathogenicity island and Salmonella pathogenicity island 2. Till now, people have identified some 13 Salmonella pathogenicity island has been identified with regard to Salmonella organisms. And these Salmonella pathogenicity islands, they are responsible for what is known as pathogenesis, as well as in the formation or the creation of what is known as all these virulence factors. So that virulence factors and pathogenic factors, they aid in what is known as pathogenesis. So like this has to be kept in mind. Like with regard to Salmonella pathogenesis, most of the virulence factors are located in what is known as Salmonella pathogenicity island, and in Salmonella, most of the virulence factors are confined themselves to Salmonella pathogenicity island 1 and Salmonella pathogenicity island 2. Coming to Salmonella in humans, like there are two different groups. One is typhoidal group. Typhoidal group, it is mainly caused by Salmonella typhi and Salmonella paratyphi. This is not a concern nowadays. Like when it comes to foodborne infection, we always talk about what is known as non-typhoidal group. They are responsible for causing what is known as foodborne infection, and they are the one that is being that is responsible for causing disease via what is known as poultry and poultry products. Like as you all know, like typhoidal group, they restrict themselves with what is known as enteric fever, and it will be characterized by all these symptoms. This is not of much importance. Whereas the important one with regard to foodborne disease is non-typhoidal group. Like in that non-typhoidal group, like they are responsible for causing what is known as gastroenteritis, and they cause other kind of disease manifestation. Sometimes they tend to cause like systemic infection. So these non-typhoidal Salmonella organisms, they have inherent ability to colonize in what is known as the poultry birds without any detectable symptoms. That means this non-typhoidal Salmonella species, like Salmonella enteritis, Salmonella typhi, they tend to colonize themselves in what is known as healthy birds, as well as in they are not exhibiting any kind of disease manifestation. Mostly these birds colonize all these organisms, and they tend to transmit the organism both by horizontal as well as vertical transmission. In poultry production, more focus is on these serotypes, namely enteritis and typhimurium, because this is the one which is having a lot of public health significance. Why? Because it tends to cause what is known as various disease manifestation in case of human beings. Like all these things, we know all these things. We have seen all these things, but the thing with regard to Salmonella is in recent times some emerging challenges are there. One challenge is what is known as biofilm. Like many of the audience will be knowing, like biofilm is a community of single or multiple species of bacteria. They are held together in what is known as a slimy complex of sugar matrix, and they tend to attach to what is known as surfaces. Like the thing is, like here with regard to biofilm formation, here the bacteria tends to adhere themselves, and they make a shield by itself with what is known as sugars. So once a shield is formed, these bacterial biofilms are extremely resistant to what is known as antimicrobials and as well as various disinfectants. Thereby, they act as what is known as a continuous source of infection in case of what is known as poultry processing plants. You may be thinking the bird is of good quality, I produce my meat under hygienic conditions, but we tend to forget about what is known as this bacterial biofilms. Why? Because once it is formed, they are extremely resistant to what is known as various antimicrobials as well as disinfectants. Thereby, that will be acting as, because these meat surfaces, these processing surfaces will be come in contact with what is known as meat, and they will be continuously, they will be your meat will be contaminated. Like with regard to Salmonella, it has been identified that these biofilms are commonly seen in abiotic surfaces, mostly in metals, plastic, as well as in glass surfaces. That means in the processing plants, Salmonella tends to form what is known as biofilm under hostile conditions. They tend to adhere themselves to what is known as various abiotic surfaces like metal, plastic, and glass surfaces. The beauty with regard to biofilm is once they are formed, these biofilm, they are very easy to remove, and they aid in what is known as cross contamination, and they continuously they will be persisting on various surfaces, and they will have what is known as significant role in what is known as transmission of this pathogen from the processing surfaces to what is known as broiling. So this is one emerging challenge which is being normally faced with regard to what is known as your salmonella organisms. And the recent problem is what is known as antibiotic resistance. As you all know, like it is not a regional problem, it is not a national problem, it's a global problem. And people have attributed certain factors like misuse, overuse, and easy accessibility of antimicrobials is the reason for what is known as emergence of all these antimicrobial resistant organisms. With regard to Salmonella, the first incidence was reported way back in 1960s, and it was seen like initially when people were having typhoid fever, like chloramphenicol was the drug of choice. When people started using chloramphenicol, people have identified there was resistance were reported way back in 1960s to what is known as chloramphenicol, and the trend is being increasing. Like many studies have shown that there is an increase in overall antibiotic resistance, which was somewhere around 20 to 30% in early 90s, but now you can see it is as high as 70%, and in some cases, it is somewhere around 100% resistant. It is being noticed to various antibiotics. Like with regard to antibiotic resistance, like people always nowadays people talk about multi-drug resistance. There is emergence of what is known as Salmonella typhimurium definitive type 104. This definitive type, it's an there is an emergence of this clone. As a result, what happened was like this clone has been distributed worldwide, and this is one clone which has been universally present in different platforms, like whether it is India or it is Africa or in America. This clone has a widespread distribution, and it is having the property of what is known as multi-drug resistance. With regard to antibiotic resistance, we have seen most of these resistance are present in what is known as mobile elements like intragons, plasmids, and or in transposons. They play an important role with regard to what is known as transmission, as well as dissemination of what is known as this antimicrobial resistance among Salmonella strains by a process known as conjugation. That means what is happening is by horizontal transfer, there is always transmission of all these resistant genes between organisms. Initially, people are thinking from one Salmonella, it comes to another Salmonella. Now the scenario is being changing. Like people have identified, like even between intra-species transmission is there with regard to what is known as antimicrobial resistance. In Asian countries, we could notice that most of the Salmonella strains are resistant to ciprofloxacin and extended spectrum beta-lactamase, which is an emerging concern. Which is an emerging concern. Like as you all know, like nowadays there is an intensive system of rearing. There is a great demand for what is known as chicken meat, and there is an increased use of what is known as antibiotic, which further aggravates the situation and makes this situation a common phenomenon in poultry and poultry products. In India, it has been identified the prevalence of multi-drug resistant Salmonella as well as the resistant to clinically important antimicrobials such as fluorochlorines, third generation cephalosporins is becoming an emerging problem not only in India, it is also a worldwide problem. Therefore, antibiotic resistance is an emerging health issue which needs what is known as a lot of attention. So till now we have seen what is Salmonella, we have seen the history of Salmonella, we have seen two emerging problems. One is biofilm formation, another one is antimicrobial resistance. So coming to the public health significance of this pathogen, like Salmonella is one of the most frequently reported foodborne disease worldwide. Like it is always a number one pathogen which with regard to what is known as foodborne diseases worldwide. In US, it has been estimated 1 million cases annually only due to Salmonella foodborne outbreaks. In Europe, it is the most common zoonosis causing foodborne outbreaks. In India, human beings normally acquire the infection by when the food is going to be contaminated with animal feces or cause contamination by other sources. Like in India, it has been very well postulated that poultry and poultry products such as egg are frequently associated with what is known as salmonellosis outbreak. With regard to enteric fever, annually there are about 2 lakhs deaths and 20 million illness occurs every year. With regard to non-typhoidal Salmonella, like nearly somewhere around 93.8 million cases is being recorded every year with 1,50,000 deaths every year. So in addition to this illness, like this morbidity and mortality, there is also increased antibiotic resistance is there. Because of this antibiotic resistance, there is an increased frequency of what is known as hospitalization, treatment failure, treatment cost. Like again, it is going to increase what is known as morbidity as well as mortality rate due to foodborne salmonellosis. So this is in general about what is known as salmonellosis. We'll move on to the next, like we'll talk about the isolation. Like isolation is very simple. Like we can go for collection of samples, like pre-enrichment, selective plating. We go for biochemical characterization. After that, we go for serotyping. With regard to Salmonella, like serotyping is done at two places in India. One in National Salmonella Center, IVRA. There they do serotyping. And there is an institute by name Central Research Institute, Kasoli. There also they go for what is known as serotyping. Because serotyping, by doing serotyping, we can very well understand what is the circulating serotype that is present in an area. Then we can go for identification of the organism by molecular method. This is how we normally go for what is known as isolation and identification of this pathogen. We'll move on to the next pathogen. Next pathogen is going to be Campylobacter. This is the general history of Campylobacter. Way back in 1886, Theodore Eustace has discovered some, has observed some non-culturable spiral-shaped organism. This organism, later in 1906, two British veterinarians, they reported some peculiar organisms in uterine mucus of pregnant sheep. Later in 1913, McFadden and Stockman isolated these microorganisms from aborted bovine fetus. Later, only in 1944, Doyle isolated a different vibrio from the feces of pigs, and he has named it as Vibrio coli. Like later, in 1963, a genus Campylobacter was first proposed. This is the brief history. Campylobacters are known to cause disease in the 1900s itself, like 1909, but they are generally recognized as a pathogen of human disease only in 1980s. 1980s, they have gained momentum. Before that, it was known as a cause for disease in case of animals as well as in humans, but it has gained momentum only in 1980s. So coming to the general classification of Campylobacter, Campylobacter comes under the family Campylobacteriaceae. There are two genus are there within that family Campylobacteriaceae. One is Campylobacter, another one is Archobacter. Campylobacter, they derive their name from a Greek word Campylus, which means backstrassen, which means rod or bent curve. Till now, there are about 28 species has been identified within Campylobacter, and 9 subspecies comes under the genus Campylobacter. Of the various species available within the genus Campylobacter, Campylobacter jejuni and Campylobacter coli are the two important things. Out of these two, like Campylobacter jejuni is the most frequently isolated organism from what is known as poultry and poultry products. General characteristics of these organisms, they are gram-negative organisms. These organisms, when two or more bacterial Campylobacter organisms, when they come together, they form what is known as a typical S-shaped or V-shaped structure, which is called a seagull wing appearance. This organism possesses a typical corkscrew-like motion, like they will be having what is known as a single polar flagellum will be there. Of all the various Campylobacter species, Campylobacter gracilis is non-motile, whereas Campylobacter chauvet, which has what is known as multiple flagella. These Campylobacter organisms, most of the bacterial pathogens, they tend to ferment carbohydrate, whereas Campylobacter is somewhat different. They have obtained their energy from what is known as amino acids and from the intermediates of TCA cycle. Most of the strains like Campylobacter, they are resistant to cephalothin, fluoroquinolones, which are the drugs which is commonly used to treat human and animal illness. Like one important thing has to be kept with regard to Campylobacter is under unfavorable conditions, these organisms has the ability to form what is known as viable but non-culturable cells. That means they go for what is known as hibernation and they remain quiescent. When the favorable condition comes, like again that organism tends to flare up. So these are some general characteristic feature of Campylobacter. Coming to the prevalence of Campylobacter, in the United States, about 1.5 million cases of Campylobacter is being recorded every year, and it has been postulated and it has been identified. Consumption of poultry meat and for poultry products, they are responsible for 50 to 70% of human Campylobacter cases. Like post-harvest broiled meat handling, processing, and consumption may contribute to 20 to 30% of human Campylobacter infection. Like Campylobacter contamination can arise from variety of chicken products from retail grocery stores, was found to be somewhere around 38%. Cross-contamination of broiler meat can also occur at home. Like people normally say under refrigerated conditions, nothing is going to happen. But the thing is, with regard to Campylobacter organisms, these organisms can also spread to what is known as other meat when they are stored in the same packaging. In India, it was noticed that the highest prevalence of Campylobacter contamination was observed in chicken meat, somewhere around 38.6%, followed by chicken intestine, it was 24%. And Campylobacter jejuni was also identified in chicken, chopping boards, knives, and feather samples, and the prevalence was somewhere around 14%. Coming to the transmission route of this Campylobacter infection, you can see these organisms. Again, these Campylobacter organisms, they also normally seen in case of healthy animals, because these organisms in poultry birds, they maintain carrier status as well as reservoir status. This organism can be excreted via feces or from the animal meat. Human being can get the infection, or it goes to the environment. From the environment, the human beings can pick up the infection. This is just to show the various transmission route of Campylobacter infection. This is the transmission cycle. You can see ingestion of this Campylobacter species by the susceptible host. There's going to be passage to the stomach and intestine. There will be colonization. After colonization, there will be cell elimination and toxin production. Against inflammatory response is there. Whenever inflammatory response is there, there will be diarrhea and there will be fecal runoff. Once this fecal runoff is there, it is going to contaminate water sources, and it will be seen in case of animals. Again, it comes to what is known as the susceptible host. This is how the Campylobacter organism is being recycled between the environment, birds, and the human beings. So this is the general pathogenesis of this Campylobacter organism. As with regard to Salmonella, we have seen Salmonella pathogenicity island is very well required for what is known as a pathogenesis. Here, with regard to Campylobacter, motility and flagella, chemotaxis, addition, toxin production, and invasion are very crucial for elucidating what is known as pathogenesis. What happens is, this organism, by the help of flagella, they tend to come to what is known as the mucous layer. Once it comes to the mucous layer, by the phenomena, something that is known as chemotaxis. Chemotaxis means it is a directed migration of a cell in response to what is known as a chemical stimulus. These organisms are in need of what is known as iron. These organisms are in need of iron. So what it does is, it tends to come to the epithelial cells. The organisms, after migration via flagella, they come to the epithelial cells by a phenomenon known as chemotaxis. Once it comes to the epithelial cells, they tend to produce what is known as toxin. So once the toxin is going to be produced, there will be something that is known as inflammation, and cell damage will be there. To counteract this inflammation or cell damage, body tends to produce what is known as prostaglandins. When these prostaglandins are produced, what happens is, automatically the cyclic AMP levels will be elevated. When cyclic AMP levels are going to be elevated, there will be imbalance of electrolytes. That imbalance of electrolyte will be elucidated in the form of what is known as diarrhea. So with regard to pathogenesis, the pathogenesis is more or less similar with regard to Salmonella, but the thing is, there the virulence as well as the pathogenic factors are totally variable, whereas here motility as well as flagella, chemotaxis are important factors which is going to aid in what is known as pathogenesis. Apart from that, Campylobacter produces a typical toxin. The toxin is known as cytolethal distending toxin. That cytolethal distending toxin is responsible for the inflammation and cell damage that leads to what is known as inflammation. Prostaglandins leads to elevated level of cyclic AMP, electrolyte imbalance that is elucidated in the form of what is known as diarrhea. Coming to the disease in humans, two kind of disease manifestation is seen in case of humans. One is gastrointestinal infection, another is extra gastrointestinal infection. Here, gastrointestinal infection means it will be restricted to only to gastrointestinal tract. Mostly, it occurs in the form of what is known as diarrhea. But here, when it comes to Campylobacter, extra gastrointestinal infection is very, very important, because many problems like reactive arthritis, Guillain-Barré syndrome, all these things are very commonly seen with regard to what is known as Campylobacter, like Miller-Fisher syndrome. All these things are the sequelae of what is known as Campylobacter. All these things happens when a person is chronically infected with what is known as your Campylobacter organisms. So the body tends to produce what is known as antibodies. These antibodies goes and act with what is known as the neuronal nerve cells. When it's going to act the neuronal nerve cells, it causes some kind of debilitating paralysis. That is what is known as Guillain-Barré syndrome. The variant of Guillain-Barré syndrome is known as Miller-Fisher syndrome, and in a similar fashion, it causes reactive arthritis. These conditions will happen in a human being only when he is exposed to what is known as chronic Campylobacter infection. So this is in general about disease in case of humans. As we have talked with Salmonella, here again there is formation of what is known as biofilm. Normally people say Campylobacter organisms cannot survive under what is known as all these metal surfaces, because they need 42 to like 43 degrees centigrade for their survival. But that is not the case, because recent studies have shown that these Campylobacter organisms, they get attached themselves to various abiotic surfaces, and thereby they will resist the various environmental stresses such as heating and chilling. And the Campylobacter jejuni biofilms are normally seen seen in various meat processing facility and on the farms, because these food processing facilities may be a perfect environment for Campylobacter biofilm formation, because it is loaded with organic material. This organic material are rich in nutrients, so thereby they facilitate what is known as survival as well as attachment of this organism. And moreover, many studies have proven that chicken juice, they also positively impacts this Campylobacter biofilm formation. So this Campylobacter biofilm formation is an emerging problem which needs a lot of attention. Again, there is a problem of antimicrobial resistance. You can see with regard to Campylobacter organism, there is a growing resistance to erythromycin, fluoroquinolones, and tetracyclines. And like in the past, drugs like fluoroquinolones have been approved for use in case of poultry, which has triggered what is known as resistance to fluoroquinolone drugs in case of Campylobacter species. In US, the prevalence of fluoroquinolone resistance was 0% in 1990, whereas it has grown up to the level of 13% in 1911. And it is still continuing, and there is always emergence of what is known as fluoroquinolone resistant Campylobacter. Nowadays, we have also noticed what is known as multi-drug resistant Campylobacter. It has been demonstrated both in animal as well as in human isolates. But this, with regard to antimicrobial resistance in Campylobacter, the prevalence is very little in countries where the antibiotic usage in poultry production is uncommon. That means more the usage, more the multi-drug resistance is there. In countries where the usage of antibiotics is less, the phenomena of antimicrobial resistance is uncommon. Coming to the public health significance of this Campylobacter organisms, as we have discussed, it is a significant zoonotic disease of bacterial foodborne infection. With regard to Campylobacter, of the total global diarrhea diseases, 8.4% is attributed solely to what is known as Campylobacter. Campylobacter infections are predominantly seen in certain age groups such as children below 4 years, and people, and in case of elderly people, that is above 75 age group, are highly vulnerable. And this organism is people immunocompromised individuals are more vulnerable to this Campylobacter infection, and this problem is more commonly seen in low-income countries. Apart from this common intestinal problem, this organism causes various extra-intestinal that we have seen, Guillain-Barré syndrome, like Miller-Fisher syndrome, and this organism is also associated with gastrointestinal infection like colorectal cancer and Barrett esophagus. So this is the public health significance of this organism. Coming to the isolation, with regard to Campylobacter isolation is very tedious, because this organism requires what is known as microaerophilic conditions. Without microaerophilic conditions, this organism is very difficult. We need enrichment, selective plating, we go for biochemical characterization, then we go for molecular confirmation by what is known as PCR. So now we have seen all these organisms. Now coming back to the prevention and control of all these organisms, there are two different strategies as well. One is on-farm measures. With regard to on-farm measures, the first and foremost thing is strict biosecurity measures. All the precautions has to be maintained. With regard to increasing the host resistance, to reduce gut carriage, we go for vaccination, or we can go for what is known as competitive exclusion. With regard to vaccination is an ideal method to prevent what is known as all these Campylobacter as well as Salmonella. But vaccination is always a failure with regard to Salmonella as well as in Campylobacter. Why? Because there is varied amount of serovars is there. Their genomic diversity is highly varied. And apart from that, the surface expressed proteins are always variable with regard to what is known as your Salmonella and Campylobacter organism. As a result, the vaccine which you are going to develop for a specific strain may not be useful for combating other strains. In such a scenario, there is always a need for what is known as a vaccine which can be used on a broader range, rather than a vaccine which can be used on what is known as for a specific group of organisms. That is why development of vaccine is still challenging with regard to Salmonella as well as Campylobacter. Their genomic diversity is highly varied. and there is a number of species are more, the number of proteins that is being expressed on the surface, it is highly variable. That is why people are still struggling to develop an effective vaccine for Salmonella as well as Campylobacter. And there is a phenomenon, something that is known as competitive exclusion. Many will be knowing nowadays people go for what is known as probiotics. So when you're going to administer all these probiotic to all these animals, all these birds, what it does is they go and sit in the various niche of the intestinal tract. Thereby, it will be loaded with what is known as good bacteria, and this harmful bacteria cannot survive. That is one, people have tried many things like lactic acid bacteria, like some yeast like Saccharomyces have been tested, and that tends to prove some kind of improvement with regard to eliminating these pathogens with regard to what is known as colonization. Nowadays, people don't advise to go for what is known as antimicrobial because there is something that is known as resistance is being developed. So people normally think of what is known as alternatives to antimicrobials. Like people go for acidification of water. Mainly, people go for organic acids like lactic acid, capric acid. We can go for all these acidification of water. Acidification of water also aid in what is known as prevention of colonization. We can go for prebiotic. Like prebiotics are the specific nutrients that will specifically enhance the growth of one organism, as by thereby the bad or the organism which has the potential to cause disease can be minimized. And people nowadays go for what is known as bacteriophages. Like bacteriophages are there, but the major problem with regard to bacteriophages is bacteriophages are highly specific. They can target only a single species that cannot be used for what is known as an array of microorganisms. That is there, and people have tried what is known as bacteriocins. People have tried. The major disadvantage with regard to bacteriocins is they are highly costly, and their preparation of bacteriocins is highly cumbersome. So these are various alternative people are trying out, and they are going to it is going to take some time to come out with a viable solution. This is with regard to various prevention and control strategies that can be handled at post-harvest or processing plants. Like here you can find slaughters, cladding, all these things. Like here the red boxes reflects where usually cross contamination is going to happen, whereas the gray boxes reflect these are places that will enhance what is known as cross contamination. Green reflects like these are the steps where we can go for what is known as intervention. So if you're going to identify all these points like very efficiently, we can go for what is known as intervention strategies. Thereby we cannot prevent, we can go for what is known as minimizing the level of pathogen in what is known as at the time of post harvest as well as processing plant. So various methods have been tried at the post harvest and for processing plant in slaughterhouses. Segregation of what is known as positive batch from negative batch, it is a fruitful method. And spraying of carcasses or dipping of carcasses with various disinfectant that has been used, like physical decontamination methods like ultraviolet rays, using of ultrasound, that is also there. Like irradiation is one of the highly effective methods, but it has what is known as poor consumer acceptability and difficulty in implementing under high throughput conditions of the major shortcoming. Freezing of carcass, that is helpful in some way. Even in spite of all these things, there is always some level of product contamination that must be tolerated. And it also varies from countries to other. In this regard, there is more emphasis on what is known as reliance of HACCP system for monitoring all these foodborne pathogens in poultry processing. Like with regard to HACCP, many people will be knowing like it is a farm to fork approach. Normally, with regard to all the existing system, we always go for what is known as end product analysis. Whereas when it comes to HACCP, like it is an integral part of food safety where we can identify hazards at various stages of processing. Like implementation of the HACCP system does not going to overcome this drawback, but number of benefits are there. Like here, the first and foremost thing is by implementing HACCP system, we are going to ensure what is known as regular monitoring of the process as a whole, and we are going to check the control parameters, and you're going to record all the results as an integral part of the system. And we always say that once an HACCP system is being adopted, we can say it is in compliance with the hygienic legislation is being ensured, and it also increase awareness among the staff. As a result, the food safety requirements will also be increased. Once you are going to implement an HACCP system, and occupational standards can be made uniform throughout the industrial level. That is the advantage of what is known as your HACCP. Nowadays, people concentrate on what is known as microbial risk assessment. Like it is a developing concept which is complementary to what is known as HACCP principle, which includes hazard identification, exposure assessment, hazard characterization, and risk characterization. Like here, we are not only identifying, we are not going to identify the risk. Apart from that, we'll be discussing the extent to which the risk can be reduced by making what is known as specific intervention strategies. By doing this microbial risk assessment, the hazard at a particular control point can be quantified with this approach, and this approach may be useful in minimizing what is known as the contamination of these pathogens. To conclude, like I'll conclude this way, this foodborne illness is a major public health concern. The burden of Salmonella and Campylobacter in both developing as well as developing areas is the constant pressure on health services because of this antimicrobial resistance due to frequent hospitalization and development of drug resistance in human. Both your poultry farms and processing systems are complex and they require what is known as intensive operation. Till date, there is no effective control method which is available to prevent these pathogen contamination, either in poultry farms or in processing plants, except for biosecurity measures. Like risk reduction strategies may be helpful. Like here, we should always think this way, like more research is needed on the application of what is known as promising intervention strategies. Like what we say is, like you go for a concept like ideal concept, synergistic action, which will eventually lead to combat these pathogens in poultry meat to ensure what is known as food safety. Rather than targeting a single control measures, a group of concepts can be evolved thereby because there is always a problem of what is known as existing problem and emerging problem. To control all these things, like a promising intervention strategies, which is going to include various methods like ideal concept, synergistic action, like has to be implemented. Then we can aim for what is known as combating these pathogens in poultry meat to ensure what is known as food safety. Thank you.
Thank you, Dr. Porteen, for the exhaustive lecture in a short period of time. Thank you so much, Dr. Porteen. Any participants, any doubt or any clarification from Dr. Porteen? If there is no question, then we will switch over to next session. Thank you, Dr. Porteen. Thank you so much.
Thank you, sir. Thank you, sir.
Huh? Yeah, we will continue with other session. Another session. You participate be in the class. இது லைஃப் ஸ்டாக் காம்ப்ளெக்ஸ் ஏதோ ஒரு மூவ்மெண்ட் ஆயிருக்கு.
Sir, can you hear me, sir? Hello?
Audible, Arthanari.
Okay, thank you. Give me share option.
So you can share, sir.
Yeah, okay. Thank you.
Dear participants, we have had an exhaustive lecture on microbial hazard, especially with emphasis on Salmonella and Campylobacter. Now here we have Dr. Arthanari, who is going to discuss about avian influenza. So kindly be attentive in the class. All the participants ask others to come and join. Dr. Arthanari is an assistant professor. He is also alumni of our campus and he is working as assistant professor in our university for the past 15 years. Presently, he is working as assistant professor in Veterinary College of Research Institute, Udmalpet. Dr. Arthanari, please take your dice.
Thank you. Is my screen visible?
Yes, it is visible.
Okay, thank you. Good afternoon, everyone. First of all, I thank the university officers as well as the organizing secretary for giving me an opportunity to present my topic in front of you. Good afternoon, one and all. My topic is microbiological hazards in poultry meat with a special emphasis on avian influenza. As you are all aware that now the respiratory pathogen which is causing havoc in around the world almost for the past 1.5 years, this COVID pandemic, we never expected that this SARS-CoV, severe acute respiratory syndrome, COVID-19 would cause this much serious pandemic and widespread disease around the world. We never even imagined that the lockdowns are still continuing. It happened in 1918, Spanish flu, where we had a similar type of pandemic. During that time, almost 5 crore people, almost 50 million people died due to Spanish flu. That is mainly swine flu, that is caused by another influenza virus, which is causing infection in swines, and it jumped to the human, and it causes serious illness, as well as it spreads from one human being to another human being. As this pandemic source, this COVID-19 has gained the potential, and it is spreading from one human being to another human being. So the whole world is being infected now. So in this context, we are also much worried about avian influenza. We are much worried about all these infectious diseases, especially this type of pandemic, starting from plague, tuberculosis, influenza, and then avian influenza, swine influenza, even we thought of, like Ebola, Zika, even Marburg disease, all these viral hemorrhagic fever, as well as acute respiratory diseases. These two are highly contagious, as well as infectious, as well as contagious, and it can cause havoc around the world, as we can appreciate the importance of COVID. So in that context, this avian influenza is also another a notorious pathogen. Still now, it had not spread from one human being to another human being. Even if at all it happened, it is of people who are with close contact taking care of the avian influenza infected people, and the viral load is very high, and the people who took care of them are under stress, and they got the infection, but mild infection. Not much mortality has happened due to avian influenza in human beings. But once it has changed, as all RNA viruses, if it spread from one individual to another individual, like COVID, and this avian influenza can also cause similar type of diseases in human beings. So we are much worried about influenza virus. It is an RNA virus. So my presentation will be in 3 parts. First part, we'll discuss about what is avian influenza. Second part, what are the OE regulations for import and export of this meat and egg products. And third one, how to handle the meat when suspected for influenza, even in a whole, how to handle the meat when there is a microbiological hazard present in the meat. So this is my plan of presentation. So we'll start here. So these influenza viruses are RNA viruses. They have 6 segments. They are enveloped, and there are 3 types of influenza virus: type A, type B, and type C. Out of which type A, which causes infection in multiple species, whereas type B, which causes mainly infection in human beings. That is common cold. That is more prevalent in Western countries. It's a seasonal cold. During every winter, people will have this type B infection, and almost every people will have must have come through this influenza occurrence in their lifetime. So it causes many epidemics, small packet of disease outbreaks. And then next comes type C. It is mainly of humans and swine. It's very rare as well. So regarding influenza viruses, they are enveloped RNA virus, and there are 16 HA types, as well as 9 NA types. HA is hemagglutinin antigen. So antigenically, it is been different classified into 16 hemagglutinin antigens and 9 neuraminidase antigen. So based on this, we can type the influenza viruses and their antigenic changes. Why we are much concerned about influenza virus? It has a potential. This is an RNA virus, similar to initially when COVID came. It was only a preliminary, genetic genotyping showed that it is a lineage of a particular origin from China. So later, due to mutation, RNA their genetic structure has changed, and now we got Delta. It is more, people say that, scientists say that is more virulent. So RNA viruses are highly prone for mutations, and these mutations are many times of not of much important, but when it happens on a particular region, virulent gene, it causes more damage to the host. So in that, we have three categories. One is antigenic shift, recombination, and antigenic drift. Shift is segment change. As I told you, RNA viruses, it has 6 segments. If segments between two different viruses, two different influenza viruses being changed, that is called as antigenic shift. Recombination, it is mixture of both, mixture of two viruses. Okay, so results in two different resources, two different viruses. Whereas drift is, this is the accumulation of point mutations of a single gene insertion, deletion, or substitution. Okay, there might be a antigenic drift that will accumulate, and it can also cause genetic change, and which will lead to a potent, more virulent virus. So these are the three categories, but most important one is antigenic shift. Okay, that is genetic reassortment, so which causes different H1, H1, N 1 to 9. So it can be of any combination. Similarly, between these, out of which H5 and H7 are avian influenza viruses, and that is, they are highly pathogenic avian influenza viruses, H5 and H7. So this is reassortment. So between H5N1 and H3N3. So if it causes, if it can, there will be reassortment. It can happen in human beings, and which lead to H5 and N3. So which is more, which causes more disease in children as well as in geriatric persons, and it can even cause death. So this is reassortment, and this is shift, antigenic shift. So these avian influenza viruses are being present in all water fowls, and there it won't cause much damage to the birds, and they are silent, and there will not be any clinical signs. The birds will not show any clinical signs, so it is unnoticed. From that birds, it can jump to any migratory birds or wild birds, or even to domestic birds, and which can in turn cause more disease in poultry, swine, or even in the human beings if it jumps to species. So we call swine as a mixing vessel. There, two different viruses from two different species will mix in the swine, and it can develop into a more pathogenic one. So that's why it's called as reassortment in swine. That's why it's called as mixing vessel. So this is transmission of avian flu. So it can either the avian flu can directly infect human beings, and from human beings, one human being to another human being. Till now, there are very few reports. It didn't jump from one human being to another human beings, and it didn't spread much. But our concern is like COVID pandemic. If avian influenza virus spreads from one individual, one human being to another human being, and another, you can expect another pandemic. So yes, this is fowl plague, otherwise called as avian influenza. It is a very infectious as well as contagious disease. It can infect more different species of poultry, duck, guinea fowl, peasants, and it can affect almost all the birds. And it causes respiratory infection as well as it causes egg production in the layers. So mode of transmission, it can directly through direct contact or droplet or aerosol route, or fecal-oral contamination, or it may be due to fomites, that is by equipment, cages, or these poultry product vehicles. It can spread through other forms. So there is a possibility of vertical transmission as well. So pathogenicity, the pathogenicity, it can be classified either it may be low pathogenic avian influenza or highly pathogenic avian influenza. Low pathogenic avian influenza in poultry, it will not cause much mortality, less than 5% only mortality. But it can, when it is being mixed with other secondary bacterial infection or other viral diseases like Newcastle disease, infectious avian infectious bronchitis, even mycoplasmosis bacterial disease, there are E. coli, it can cause severe infection, and there will be mortality will be higher. And highly pathogenic avian influenza viruses can cause mortality up to 100%, and it is a notifiable disease. It has to be informed to the OAE by the concerned government when there is an outbreak. So low pathogenic form, it is very common, not common. The severity infection is very less. Mild respiratory signs only, ruffled feathers or reduced egg production, and this is a major problem in laying layers. It can cause a drop in egg production, and it can the infection is mainly through migratory birds or other waterfowls, and the disease can spread when from one place to another place when due to live market or backyard poultry, and it can mutate into highly pathogenic form. So these are the low pathogenic avian influenza lesions. So you can find petechial hemorrhages on pericardial fat, and you can, similar to Newcastle disease, you can find proventricular papillae hemorrhage and petechial hemorrhages on the fat, and then peritonitis, egg peritonitis. The bottom one, right hand side bottom one is egg peritonitis. So, and next one is highly pathogenic avian influenza. This is more dreadful, and if it happens, the samples has to be sent to Bhopal, and then this causes mortality up to 100%. The only problem is when there is an highly pathogenic avian influenza outbreak due to H5 or H7. Okay, so the birds has to be culled. It has to be stamped out because it can spread very in a fast manner, and it can cause havoc in the poultry industry. So it, as well as it is of more zoonotic potential, so the birds has to be stamped out. Okay, so the lesions you can find sudden onset, respiratory illness, multiple organ failure. You can find hemorrhages on all over the visceral organs, and you can find bluing or discoloration of cyanosis of shank, and it can cause rapid spread, and mortality can go up to 100% within 48 hours. Clinical signs, the main it varies with age of the bird as well as method of management, environment factor, and we have many issues. We are not much concerned about clinical findings. We'll go to lesions we have already discussed. You can find egg peritonitis, and you can find hemorrhages on epicardial fat and abdominal fat, and you can find hemorrhages in the gastrointestinal tract from the gizzard, proventricular gizzard to you can find up to cloaca. You can find many changes, necrotic foci and visceral organs, and kidney. You'll find urea deposition in the. Then diagnosis based on the history and as well as postmortem lesions, and the gold standard method is isolation. But as soon as we send the samples to the cloacal swab or respiratory swab, we collect and we send it to Bhopal. They do reverse transcriptase PCRs, polymerase chain reaction, to detect the viral RNA, and later they go for isolation, and they do typing. Based on typing also, they say that whether it is H5 or H7 and what is the N type. So they do hemoagglutination inhibition test. We have ELISA, FAT, AGPT, and treatment, as you all are aware that for avian influenza, you can go for Tamiflu or Oseltamivir and amantadine hydrochloride in drinking water. This is as well as it is being used in humans as well. So control, mainly stamping out, disposed, all the birds should be culled, and the premises has to be completely cordoned, and it has to be disinfected. Dead birds has to be properly burnt or deep burial, and all the materials which are present in the farm should be properly cleaned and disinfected, and there should be a strict biosecurity measures. Even the people who are working there should be monitored for at least for 15 days to see is there any disease in the human beings, because as it has a zoonotic potential, we are much worried, and the virus can stay in the feces for quite longer time, almost for 3 months, and if it is being refrigerated, it can survive for quite long, longer period. And the virus will be, before death, the virus will be excreted from the bird through feces. So feces is the main source of viral virus. So the complete farm has to be sealed and properly disinfected, and then there should be a proper downtime before introduction of new birds. Okay, so they can be easily destroyed by our sodium hypochlorite, formalin, even Vircan, which can also easily applied, and you can completely kill the virus. Okay, these are the previous pandemic. This we have already discussed, and this is the report from OIE. Every year, OIE will give a notification about occurrence of avian influenza twice every 6 months. They update. So this one is being released in between December to January. So there were almost 250 outbreaks in the last year. So even it had happened in India as well. So these are the types you can find, all mainly of H1, H5. Almost all are H5. So it was being reported from Africa, Asia, as well as Europe. You can find how many number of outbreaks and how many number of birds infected. These are the spatial distribution of the outbreak. So around 250 new outbreaks. So the total HPI outbreaks worldwide is around 372. All these blue dots on the map, and red dots are from that is from Africa, Asia, and Europe, and rest of the 213 from Africa, Asia, and Europe. They are the blue, red ones are the previous quarter. The blue ones are the present one. So next one is as avian influenza is highly contagious, as well as it has a high zoonotic potential. As soon as we come across H5 or H7 avian influenza outbreak, it has to be notified to OIE, World Health Organization for animals. So as well as nowadays, even the low pathogenic viruses, like H9 and other subtypes of H7 and H9, if it has potential to cause infection in human beings, if it is being reported, the people, when bird handlers are infected with low pathogenic virus and if it causes severe illness, even that has to be reported. Not only the highly pathogenic avian influenza, even low pathogenic avian influenza, when it has capacity to infect human beings, it has to be reported, because the genetic nature of the viruses keep on changing. Even the low pathogenic virus can mutate, and it can even can cause a serious illness in human beings, and it can cause even pandemic. So that's why not only the highly pathogenic avian influenza, even low pathogenic avian influenza virus has to be reported nowadays. So this animal to human transmission, that is from bird to humans, mainly people working in poultry farms, veterinarians, as well as even slaughterhouse people are highly risk individuals. So they should have proper PPE, personal protective equipment, when they handle these infected birds. Even if you go for stamping out, the people should be of even sampling or stamping out, or even just to collect the samples also. People has to be highly cautious. They should have personal protective equipment, and then only they can handle the birds. So we are much worried about it should not turn into a pandemic one. That's why it has to be properly there will be a continuous surveillance of avian influenza virus throughout the world. Even in India, regularly, from starting from northeast to our Tamil Nadu, Kerala, even Rajasthan, Punjab, all migratory birds as well as poultry samples regularly sent to Bhopal for screening. What type of virus is being circulating? So continuous surveillance is there, both of mainly of RT-PCR method, molecular method. And if birds from if we can collect serum, you can even go for ELISA and see the subtype prevailing. And when there is a human infection due to avian influenza, so it can cause persistent fever, lymphopenia, and ARDS, that is the acute respiratory disease syndrome. Like COVID, you will find all similar clinical signs, starting from fever, body pain, and there will be sore throat, coughing, sneezing. So you can find joint pain. So like all other viral diseases, it can cause serious illness, and it can be treatable. But even the person who are accompanying the sick person has to be very careful. And that's why, because the infected individual, normally you can say it's a dead-end host. Humans right at this moment, they are a dead-end host, but there are possibilities if the viral load is very high, and the person who are handling the infected person can also get the infection. But still now, very few instances had happened. Otherwise, up to now, there is no problem of avian influenza in humans. It didn't cause any pandemic. And as far as foodborne public health significance due to foodborne pathogens, this virus normally it is being present in respiratory tract as well as gastrointestinal tract. But when there is a highly pathogenic virus, and when the bird is and with severe infection, the virus can reach any place. So it will be there in the blood, muscle, it will be everywhere throughout the body. So even it may present in the meat as well. But the viral load may be very less, or when you handle many times, we don't get the infection. But it doesn't mean that it will never cause infection. It can anytime, depending upon the individual, or depending upon the amount of virus present in the bird, or the health status of the person who are handling the infected meat, can get the infection. So it depends on various factors. But always, treat this pathogen as a highly potential, an occupational hazard or foodborne pathogen. So you have to prepare yourself and then handle the infected meat as well as egg. But as it applies to all other foodborne pathogens, these even viruses, it can be easily inactivated at the temperature of 75 or 70 above 75 degrees centigrade. So it will easily get inactivated. So there are it can be easily processed. So when you handle, you have to be very careful. So with all personal protective equipment, you have to handle the infected meat. Better avoid getting meat from infected area, or even from live market, where the high possibility of getting the infection. So these are the different temperature, thermal, death time for this virus. It literally inactivated at 73.9 degrees centigrade within 0.5 seconds. So at 70 degree, it's 3.5 seconds. So these are the thermal death time. So this is the OIE FOO site recommendation summary notes. When there is an outbreak, what we should do better, the best is cull the birds, stamping out, complete stamping out. If at all, if it escapes and if it reaches the market, or if it is been because in developing countries always the problem of compensating the farmers with a lump sum amount. So there are tendency that the farmer may sell before the authorities go and do stamping out. So even if it reaches the food chain, what are all the precautions we have to follow? Better not. You should not take any. The bird should not be sent to the market at all. It has to be completely destroyed. But freezing and refrigeration can the virus can survive in the meat as well as on the eggs. So only by cooking, you can completely inactivate the virus. There is no problem of eating completely well cooked meat as well as egg. Even egg, it should be completely cooked. It's not half boiled or partially boiled. So it should be completely cooked and consumed. So these are the notes being given by OAE for when there is an avian influenza outbreak, what are the food safety implications. Okay, so home slaughtering and preparation of sick and dead bird poultry is hazardous, so avoid. Don't do processing of chicken when it is bird is from the suspected outbreak, so it should not be slaughtered. And it should be completely cooked, and if at all, if it reaches, it should be completely cooked and being consumed. So live poultry, these are the recommendations for import or export of poultry. Always we have a policy as far as avian influenza is concerned. It is being divided into zone, a compartment, zone, and then country level. Compartmentalization of a particular establishment, a farm or group of farms, if they have a proper biosecurity measures as well as surveillance, that is those birds are monitored regularly at regular intervals. If it is being monitored for avian influenza presence, if it is negative, that area can be a compartment. From there, you can export or you can transfer the birds to any country, because that is a avian influenza free compartment. Zoning, a huge area where the group of poultry farms or group of establishment are free from outbreak. So that is called as zoning. And country, as you know, that regular surveillance and screening you can also show that, you can also prove that your country is free from avian influenza. So in that case, you can certify that these flocks are monitored and found they are negative for avian influenza, and you can export the live bird or eggs or even the meat to any country. So it depends. The policy of importing varies from country to country. US has a different standards. European Union has different standards. African countries, Middle East, they have different standards for importing meat or eggs. So depending upon the country, the necessity, the rules, regulations varies. But even within India, if the particular region is having infection, whereas if Namakkal is free of avian influenza, you can export. Okay, if that particular country agrees that Namakkal is, if you can substantiate with your all proper surveillance document, you can export the poultry from the avian influenza free zone. So that is regarding live poultry. It applies to even meat as well. Meat also can be exported to any country if you can establish a avian influenza free zone. So these are the recommendations for poultry meat. So it is similar to live bird as well as eggs. We have to establish that our region is free from avian influenza, and we have to certify that these flocks are being regularly monitored for presence of avian influenza and it is negative, unfound negative. And eggs for consumption, as we have already discussed, similar to meat as well as live bird, even eggs, they have to be from the avian influenza free flocks. So it has to be, veterinarian has to be certified through all government agencies only. People, they importing countries ask for standard documents from the government, not from individual players, but individuals has to approach the government and get the certificate and then proceed for export. So like a safe commodity, even if it is being processed meat product or pet products, when it is being treated above 75 degrees centigrade, it can be considered as completely inactivated, viral inactivated products, and there will not be any possibility of spread of the infection. So even meat meal, blood meal, bone meal, and feathers, even pet foods, pet foods made from offals or other meat products, all should be properly packed, and if it is being properly processed, it can be exported. So they are safe commodity. And now we come to the last part. What are the guidelines for handling and consumption of poultry meat during bird flu outbreak? So this is being given by FSSAI, Food Safety and Standards Authority of India. It is being published in the net. You can download it. They have given separate guidelines for meat products, eggs, as well as food consumption as well. So so precautions taken at the retail meat shops. So it almost the processing, handling of live bird, as well as slaughtering, and as well as preparation or cooking the meat. Wherever we handle the live bird or meat, if it is being suspected, normally it should not be consumed. As I already told you, avian influenza outbreak, the birds from the avian influenza outbreak area should not reach the food chain at all. Stop it. It should be completely banned. Even if you suspect it is a neighboring area, if you suspect, but the birds are not having any clinical signs, and even maybe some reports also, you can say that some farms might have tested their birds, and they might have got a negative result. So if it reaches the food chain, whatever the origin of meat, always handle as a cautious one. Which is having a pathogen. So you have to prepare, use all personal protective equipment like headgear, mask, goggles, gloves, as well as apron and shoes. You have to completely cover yourself and then handle the birds during handling the birds, as well as slaughtering and preparation. Even the materials which we are using, it has to be properly washed, then disinfected, and then it has to be used further. Even the material which is being disposed, like offals, other things, has to be properly decontaminated and disposed. It should not be thrown as such outside. And it has to be handled very cautiously, because even these aerosols can cause infection in human beings. But as for now, we never come across that much serious illness due to avian influenza. Even if it all happened, till now there is not much reports of human, very virulent avian influenza which is causing human infection or human mortality. Of course, we have found almost around 350 to 400 deaths almost for the past 2.5-3 decades. We recorded death due to avian influenza, but not of very few incidences. They might be having some other comorbidity, and the viral load is very high. Due to that, the mortality was, reported. Otherwise, till now, it may even, though they are highly potential hazard, still we are lucky that it didn't turn like a COVID pandemic. Okay, so but even then we have to handle the meat in such a way that it has a pathogen. So next is by consumers, avoid live market purchase during outbreak. Avoid collecting birds from the infected region. Even if you have bought live fresh meat, it has to be properly handled, like what we do it in the slaughterhouses. Similarly, it has to be handled in the house. Even before cooking, it has to be the vessels which are being used should be properly cleaned and then dried completely. Even the material, the knives or cutting boards, whatever it is, it has to be properly cleaned and disinfected. Even the water which is being washed away in the drainage, it has to be properly decontaminated. It should not be left as such in the open drainage. It has to be decontaminated and then disposed of. So even cooking the meat, as I already told you, above 75 degrees centigrade, the virus is being completely inactivated. So even egg has to be completely boiled, completely cooked and consumed. You should not use half-boiled preparations for any of the dishes. Even egg or meat, it should be completely cooked and consumed. And even handling also, you have to protect yourself. These are the some USDA standards. USDA also say that the virus getting, inactivated at above 74 degrees centigrade. So eggs also we have already discussed. Similar to meat, egg also has to be properly handled, even because the virus is being excreted through the feces. If the egg surface is contaminated with or stuck with some feces material, it has to be handled as a potential viral source. Okay, so it should not be washed as such in the running tap water, because the splashing of the water droplets may contain virus and it can easily contaminate the environment as well as the handler. So it has to be wiped, or it may be cleaned with a towel with a formalin or a good disinfectant, and you can use it for you can transport, or you can send it to the consumer. But vertical transmitters, there is very few reports, not much of worry. As I told you, it has to be completely cooked and consumed. So that is most important. So these are the some do's and don'ts while handling meat, bird, as well as eggs. So half-boiled, it should be completely stopped. So undercooked also not advisable. Avoid direct contact with live birds from the infected area, because it is not possible, because now we have good awareness. As soon as there is a report of outbreak, the whole area is being completely guarded, and there is no escape of birds from the infected area to the surrounding area. So there is no possibility, but in far remote villages, there is possibility of birds may be transported to the new area. So that should be avoided. If you say that a particular region is having outbreak, you better avoid getting birds from that and consuming. And avoid touching dead birds with bare hands. As I told you, even whoever it may be, when there is an avian influenza outbreak, it has to be like what we are seeing in COVID. COVID patients, how they have been taken to the hospital, or even when treating also doctors as well as attenders, nurse, whoever it may be, they will be under, with complete PPE kit only. So similarly, even the dead birds, or collection of dead birds, or disposing the dead birds, or even when people go for blood collection or collection of swabs, cloacal swab or nasal swab, during that time also people should be in PPE. So don't keep raw meat in open, as well as no direct contact with raw meat, and always handle the suspected material as a potential pathogen. So personal protective equipment, now we all know what are the personal protective equipment. Initially the recommendation for COVID is people should wear N95 masks, later surgical masks. Now we have many varieties of many different materials, but later we have come down to that if you close your mouth and nose, that is sufficient. We never bothered about whether virus is being respirated through or respirated in. So we don't worry about the whether virus can pass through or not. We simply wear a mask, that's all. That itself, now it is mandatory and we are following it. Actually speaking, it can reduce, but it will not completely protect. Yeah, whereas the N95 can completely protect, but the infection can go through eyes as well. So even respiratory route or oral route, or through conjunctival contact. If after handling the infected material, if you rub your eyes or nose, so you can pick up the infection. So there is a possibility that virus can transferred from the environment to the host. Okay, so there is a high possibility. So you have to be well prepared while handling the meat or the birds or even the eggs. So these are the work practices, we have already discussed. What are the work practices? Always consider this as a potential pathogen. So you have to protect yourself. You should not eat, drink, or even rubbing your eyes. That should not be done while handling these suspected materials. Even if it is any material, if it is a source of this hazardous avian influenza, not only avian influenza, we have many foodborne pathogens which can even cause diseases in human beings. Even if it is from a good, well-known, farm, without any, if it is not even having any contaminated with any of the zoonotic pathogen, always handle hygienically. That is the most important. So when you start maintaining hygiene, both of personal as well as the material you're handling, it easily you can prevent yourself from exposure to the pathogenic organism. So avoid direct contact with the contaminated material or secretions, and it has to be reported. Even the farms or the area which is being exposed to the virus, even if you are slaughtering the bird, or if you are handling, or if you are cooking the meat, the materials which are used for cooking, the cooking vessel with meat will reach the very higher temperature because you are cooking, so virus will definitely get inactivated. But the chopper board or the knives or other utensil we use it for preparing the meat or processing, that has to be properly cleaned and disinfected. That is more important, because after again cooking, if you are transferring to the same vessel, definitely the virus will be there. Only the meat, cooked material is devoid of the virus, whereas the vessel will have the virus in it. So always use a fresh vessel, or it should be properly washed and cleaned and then used. So don't use the same material with the same, which is being exposed to the raw meat. So that is most important. So these personal protective equipments, you are aware what are all the personal protective equipment, starting from headgear, goggles, mask, then overall, there are good quality gloves. Even if it is been torn or there is any tear, it should not be used. You have to change the gloves, because there is high possibility of the virus can reach, even if it may contaminate your fingers, because due to the damage of your gloves, even your fingernails, finger can get contamination. After removing PPE, you have to take shower, or otherwise you have to maintain hand hygiene. Otherwise, even if you touch the infected meat and if you rub your eyes, that is sufficient. Even theoretically, if is hypothetically, even a single virus can cause a complete infection. But we always talk, that the amount of virus present in the secretion or the feces, that is, we always say that it should be more than only the individual get infection. But theoretically, even a single virus can cause disease. Okay, so always handle them very cautiously. So these are some. It's all downloaded only. You can even now we are everywhere. The whole world is aware of what is PPE, what is N95, what are the protective goggles, and what is the difference between surgical mask and N95. We have many other gaseous chemical respirators as well. But this is for microorganisms. So then aprons, gloves. This is for filter respirators. So these are not necessary, as even if it is an outbreak and you are handling more amount of birds and other things, definitely these type of respirators are needed. Otherwise, it is not necessary. And hand hygiene, like what we are following now, social distancing, mask, as well as hand hygiene and hand sanitizing. So minimum of 20 seconds. Completely, we have a procedure to clean our hands, but people are just rinsing with water and just a drop of soap. Within 5 or 10 seconds, they complete their procedure. But actually speaking, you have to wash your hands at least for 20 seconds with soap, and then completely rinse it with running tap water. Then only you can say that you have done a good hand hygiene. You are doing hand hygiene. Otherwise, it is not considered as hand hygiene. So be cautious. But we have gone through the whole. This process for the almost past 1.5 years. Everyone is aware of it. But similarly, this applies to avian influenza outbreak as well. So as far as what are the disinfectants we use it for cleaning and disinfection of the premises, or the utensils, or the farm, or the cages. What are the disinfectants you can use it? This 2% sodium hypochlorite, 4% formalin. 2% formalin is sufficient. If the load is very high, you can go for 4% formalins, and 3% calcium hydroxide, and you can use potassium permanganate and formalin fumigation. If it is a closed area, you can use fumigation. And cages and other things and feathers, you can use flame guns, and you can completely destroy the feathers as well as virus which is present in the cages. And it can be, after that, it has to be completely washed and disinfected. Again, you have to spray it with sodium hypochlorite or formalin. Again, it has to be after washing, you have to again clean it. Okay, and then you can use good virucidals like Vircon. We have many other branded products available in the market, so you can use any one of these. But sodium hypochlorite and formalin works very well. And I think, okay, that's all. I think I have covered within a short period of time. I could able to cover the importance of avian influenza, and if it reaches the food chain, what should be done. Thank you. Any questions? The session is open for questions. Sir, am I audible?
Yes, sir. Audible. Participants, if you have any question, you can ask, sir. Or else you can type it in the chat box. No
So
question.
no questions, sir.
I think, sir, no question, sir.
Okay, thank you, doctor.
Thank you, sir.
I have given my number as well as mail ID. If you
Okay.
have any other queries, you can contact me.
Okay, sir. Okay, sir.
Thank you, doctor. Thank you.
Okay. So on behalf of participants, I thank Dr. Arthanari, sir, for his wonderful presentation. Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir. So participants, thank you. And next session will start by 3 o'clock, and we can wind up this session. We will join in that link that is given for 3 o'clock. Thank you.